Bye.
Okay, so we are going to finish with chemical digestion, and then we're going to talk about metabolism as well, but that will be a separate Zoom. All right, so we left off with digestion is talking about how we're going to have to hydrolytically break down each organic family to their building blocks. Let me just make sure that let me just reestablish my chair just to make sure we're in the right one. This one. Okay. Okay. So let's look at each organic family individually to identify how we're going to break it down, what enzymes are going to be used, and where those enzymes come from. So digestion of carbohydrates begins in the mouth with salivary amylase. So it starts to break down a polysaccharide to a disaccharide. However, when you swallow food, it travels into the stomach, and now it's going to come in contact with hydrochloric acid, which then stops any further or the majority of any further digestion of that polysaccharide to a disaccharide, because the hydrochloric acid there denatures the enzyme, the proteins of that enzyme. So that's why we keep saying we begin carbohydrate digestion in the mouth, but it's not completed. So now when we gastric empty and these components partially digested, partially broken down, polysaccharides and some polysaccharides that haven't been acted on at all, we need to reinitiate that again. We need to get everybody down to the disaccharide level. So now as it leaves the stomach in chyme and enters the small intestine, the pancreas will release pancreatic amylase, and that will finish breaking down those polysaccharides to disaccharides. All right, so now everybody theoretically is a disaccharide, and the disaccharides are sucrose, maltose, lactose. So now as the food propels farther down in the small intestine and the conveyor belt's moving farther down the small intestine, it passes the brush border cells producing those brush border enzymes. So now we have secreted lactase, sucrase, and maltase. Sucrase acts on sucrose to break it down to glucose and fructose. Lactase acts on lactose to break it down to glucose and galactose. And maltase acts on maltose to break it down into two glucose molecules. So you should you should know what the substrate of lactase is. That would be lactose. And what are the products of digestion of lactose? One glucose, one galactose. You should know for each one. Sucrose, the end products of digestion of sucrose is glucose and fructose. Of maltose, it's two glucose molecules. And these enzymes come from the small intestine. Pancreatic amylase comes from the pancreas. Salivary amylase is produced by the salivary glands in the mouth. So that's how we break down our carbohydrates. Right, then we look at proteins. Proteins is a little more complicated, so just bear with it all, right? Because the last two families are really easy. But protein digestion is more complicated because you have very large, complex, molecules with a primary, secondary, tertiary, quaternary structure. They're held together by hydrogen bonds, but those hydrogen bonds are weak. So protein digestion begins in the stomach. Part of those hydrogen bonds are broken down by the hydrochloric acid there. And once we start to unravel all those proteins, then pepsin can start jumping in and cleaving, those polypeptides to dipeptides. Polypeptides to dipeptides. Pancreatic enzymes. So now we're going to, again, just like the carbohydrates, some of those proteins have been broken down to dipeptides. Some of them have not. So now as that, partially, or some of those proteins that have not been broken down yet, as it empties into the small intestine as a component of chyme, passes the pancreas releases trypsin, chymotrypsin, and carboxypeptidase. The trypsin and chymotrypsin, primarily act on the polypeptide to break it down to the dipeptide, and the carboxypeptidase is really cleaving off that carboxyl group at the end of the molecule, that COOH group. But again, it helps break it down to dipeptides. And now that they're all essentially at the dipeptide level, and that conveyor belt moves farther down along the smooth, the small intestine pathway, it passes those brush border cells that produce the brush border enzymes. And now those enzymes or those cells produce dipeptidase, and dipeptidase completes the breakdown of dipeptides to amino acids. Lipid digestion predominantly occurs in the small intestine. We do know that there's a little bit of lingual lipase, right, in saliva, but the majority of it is going to come from the lipase from the pancreas. Bile salts, that are a component of bile, right, help to emulsify fats to increase the surface area, and takes a large molecule of fat and breaks it down into little droplets. And now the lipase from the pancreas can digest or break down the lipids into triglycerides and into fatty acids and glycerol. So it's going to break down the triglycerides to fatty acids and glycerol, and now be absorbed in the small intestine. Nucleic acids are also very easy, occurs in the small intestine. Again, we have the nucleases, deoxyribonuclease and ribonuclease. We just shorten that up to DNAs and RNAs. Are going to act on DNA and RNA to break them down into the nucleotides. And now those can be absorbed. So the nucleic acids are pretty straightforward, and the lipids are pretty straightforward. It's a one-step process. All the enzymes come from, from the pancreas. But I must admit, the carbohydrates and proteins, a little more complicated. Carbohydrates start in the mouth, and in the small intestine, and proteins start in the stomach and end in the small intestine. Okay. Absorption of these molecules. Well, most absorption occurs before it even reaches the ileum. Ileum absorbs bile salts and recycles them to the liver, so that kind of recycles those bile salts. Monosaccharides, amino acids, nucleic acids are all absorbed and transported in the blood supply of the capillaries in the villi. The fats will be transported in the lacteals and enter the lymph system. And the lymph system, of course, rejoins the cardiovascular system in the subclavian veins, right and left. Okay? So all the best way to describe it is all the molecules that are polar covalently bonded will travel right into the bloodstream, in the capillaries that supply the villi, because plasma is 90% water, so they need to have compatible bond types. And the fats will be transported in the lacteals, the lymphatic system, and then they will join the cardiovascular system later at the subclavian vein level. Vitamins and electrolytes and some water is absorbed in the large intestine. Absorbed nutrients, water, and vitamins enter the blood via facilitated diffusion, transported by mesenteric veins to the hepatic portal vein and the liver for processing. And that's the end. There's not that much more, there wasn't that much more in the chemical digestion. And I will put up, there is up on the course page, a chart, that I use in AMP1 for the chemistry of a chart that outlines the 4 organic families and the different levels of macromolecule, intermediate molecule, if there is one, and the building blocks, of which all of them, every organic family has a building block. And I use the same chart I use in AMP1, but I add arrows from the large molecule to a smaller, to the intermediate molecule, to the building block in the case of carbohydrate and protein, and in lipids and nucleic acids, we go from the macromolecule to the building block directly. And over the arrows, I write what the enzyme is, and then I color code the source. So all the pancreatic enzymes, are one particular color. The one salivary amylase is another color. though the small intestine enzymes are a different color. The stomach enzyme is another color. So I take each family in vertical columns. We identify the building blocks, intermediate macromolecules, and then identify which enzymes facilitate, the hydrolysis of those molecules into smaller ones. Identify the enzyme and color code it by source. And that's a good one to practice, over and over again. So you should find that in that same region, on the course page of in the where the digestive system notes are, and where the quiz is. All right, the next lecture will be on metabolism.